Good morning. In this video, we are going to pursue our discussion about the factors that affect enzyme action. And this time, we will be talking about allosteric inhibitors and allosteric activators. But before discussing allosteric inhibitors that inhibit enzyme action and allosteric activators that activate or increase enzyme action, we have to talk first about allosteric enzymes. What are allosteric enzymes? The word allosteric means able to change conformation or capable of changing shape. So an allosteric enzyme and an allosteric protein in general is a protein that is capable of changing its shape. How can allosteric enzymes change their shape? What do they have in particular that allows them to change their shape? This is the fact that they have two sites. They have the original active site, and in addition to this, they have another active site, or another site actually, on another part of the molecule. And this is the active site. Suppose this is the active site that is supposed to bind with the substrate. So this is the first site, which we call the active site. But the second site is present on another place of the molecule. We don't call it an active site, but we call it an allosteric site. So the allosteric enzymes are characterized by having two sites. The active site that is supposed to bind with the specific substrate, and of course that active site should be complementary in shape and complementary in charge. And another site called the allosteric site. As the name indicates, this allosteric site is supposed to be like a turn-on, turn-off button to change the shape of the enzyme. So this allosteric site will be responsible for changing the shape of the enzyme. But why do we want to change the shape of the enzyme? It depends. It depends upon the molecule that will bind with the allosteric site. That molecule that binds with the allosteric site is not the substrate. That molecule is the turn on or turn off button. That molecule is called what? That molecule is called an allosteric effector because it binds with the allosteric site and has a particular subsequent effect on the whole enzyme. If the allosteric effector is a turn-on button, or if the allosteric effector is an allosteric, that's why we give the molecule the general name of effector, because if the allosteric effector can be an allosteric activator that will turn on enzyme activity, that will turn on the enzyme activity, or it might be an allosteric inhibitor that will turn off the enzyme activity. But how can it turn on or turn off, and how is this related to change in shape? So it all depends upon fitting. So we will read first what's on the slide. An enzyme molecule, an allosteric enzyme, has a second site in addition to the original active site called the allosteric site. And the molecules that can bind with this allosteric site, as we said, are called the allosteric effectors. And this is the general name for the molecules that can fit with the allosteric site. And upon binding with the allosteric site, they change the shape of the active site. So these turn on, turn off buttons will turn on and off according to how they change the shape of the active site. If the molecule that binds with the allosteric site here is an allosteric activator, then it will make the active site more fitting with the substrate. But if this molecule that binds with the allosteric site makes the active site less fitting with the substrate, then we call it an allosteric inhibitor. And we will see on this slide the types of allosteric inhibitors and how they achieve this kind of inhibition. Of course, I remind you that we are discussing the factors affecting enzyme activity. So we talked about pH, number 1. We talked about temperature, number 2. And the effect, of course, of pH and temperature was variable according to the value of temperature or the value of the pH, or according to the degree of temperature. And number 3, we talked about 2 types of enzyme activators, which were the coenzymes, organic and large molecules like vitamins, or cofactors, small and inorganic molecules. And both coenzymes and cofactors were enzyme activators. The 4th factor that we are discussing today are the allosteric effectors. And the effect of the allosteric effectors is variable according to the type of the effector. The allosteric effector can be an activator or it can be an inhibitor. According to what? According to its effect on the shape and charge of the active site. If they become more favorable to what? To the original specific substrate, then it will be an activator. If it becomes unfavorable, and the enzyme is no more capable of binding, then it is an inhibitor. And actually, we can say that these allosteric effectors work only on allosteric enzymes. The enzyme, so they work only on allosteric enzymes. If the enzyme is not allosteric, meaning that the enzyme does not have a second site, which we call the allosteric site, then the allosteric effector can no more carry out its function. Now, we have actually 2 types of allosteric inhibitors. Type 1 is easy to understand. So here, again, this is the normal reaction. We have this green molecule that is the famous original substrate we talked about, and then it binds with the active site and gives rise to the substrate, it gives rise to the products. So we have here the reagent, the specific reagent, which we call the specific substrate, and it binds with the active site. And we notice that there is another site on the molecule that is the allosteric site. But we don't need, in every reaction, it's not necessary for an allosteric effector to bind with the allosteric enzyme. But what happens if an allosteric effector binds with the allosteric enzyme? In such a case, as you can see, when this dark green molecule that I'm pointing at with the red arrow binds with the allosteric site, if you can see it, this allosteric effector is bonded to the allosteric site, not to the active site. But it has a remote effect on the active site. How? It made the active site less favorable to the substrate, less complementary to the substrate. And now the substrate is no more capable of binding with the active site. It's not because there is a molecule that is binding to the active site. No, it's because of the remote effect of the allosteric inhibitor that does not bind and never binds with the active site, but through its binding with the allosteric site will prevent the original substrate from binding with the active site, just because the active site is no more triangular, it's rather square, and therefore it can no more be complementary to this pointed part of the substrate molecule. But this is not the only mechanism of allosteric inhibition. There is another mechanism of allosteric inhibition. Again, the reaction on the top of the diagram is the usual reaction, the normal reaction, the turn-on reaction. But here we have an allosteric effector. This is an allosteric effector, and I can jump to the conclusion that it is an allosteric inhibitor. Why? Because at the end, I found out that the reactant or the specific substrate did not change, did not result in the production of a specific product. But the mechanism is different here. The allosteric inhibitor was bonded to or bonded itself again to the allosteric site. It never, by the way, binds to the active site. And this time it did not actually change the shape of the active site, but it covered the active site with part of its molecule, and it also resulted in the prevention of the substrate from binding with the active site. So these are the two types of allosteric inhibitors and the two mechanisms of allosteric inhibition. So if I ever ask you about the mechanisms of allosteric inhibition, so I am actually asking you to explain what happens in both cases. On the other hand, as we said before, the effector might be a positive effector. A positive effector means an allosteric activator. Again, why did I call it an allosteric molecule or an allosteric activator? Because the enzyme has two sites. The first site is the active site, and the second site is the allosteric site. The allosteric site. We don't call it a second active site, okay? Here we can see that the allosteric effector, the yellow molecule, has bonded to the allosteric site, and after having bonded itself to the allosteric site, the shape of the active site is now more fitting, as you can see, with the substrate and more favorable. That's why we call it allosteric activator. Now, we have talked for long about the activation energy, and I remind you of the definition. The activation energy is the minimum energy that is required to start a chemical reaction. So we have to use these two words. It's not the energy needed for a chemical reaction. This is too inaccurate. We have to say that it is the minimum energy that is required to start a chemical reaction. So here on this slide, we are going to see how enzymes lower the activation energy, because we said before that enzymes speed up chemical reactions, but how do they are biocatalysts. We said before that enzymes are biocatalysts, meaning that they speed up chemical reactions inside living cells. They speed up the chemical reactions. And we said that they speed up the chemical reactions. How? By decreasing or minimizing the amount of activation energy needed to start the reaction. So in order, or instead of waiting for a long period of time until we reach the amount of energy needed, just a small amount of energy is required to start the reaction. So it's understandable that by lowering the activation energy, we are also lowering the time needed for the reaction to start, and therefore we are speeding up the chemical reaction. And we're going to see this on the graph. And of course, we said something important before looking at the graph, that if we have an enzyme like this that has two active sites for two substrates A and B, then the enzyme speeds up the chemical reaction and lowers the activation energy because it approximates the two molecules. It allows the two molecules to get closer to each other. And when they get closer to each other, there is an increased likelihood or possibility of collisions between the molecule and in the proper orientation or in the proper direction. And this, of course, facilitates chemical reactions because if there is an increased possibility or likelihood of collisions, then there is an increased likelihood of bonding and breaking down bonds and forming new bonds. Now, how can this be applied on the graph, which is a very important and famous graph? Here we have the reaction progress on the x-axis, and the reaction progress, of course, is time or occurs along time. So if you want to add on the graph, to label the graph with quantity or something that's measurable, then we have time, maybe in seconds, maybe in minutes, according to the rate of the reaction. And on the y-axis, we have energy. Of course, here there is Gibbs free energy. You needn't memorize the name Gibbs, but the free energy is the energy that is available for the molecules to react. So you just write energy. You needn't even write free energy. This is a bit beyond the scope of your curriculum. And here we have the reaction. The blue curve represents the change in energy along time or with time. So at the beginning, we had the reactants, and then we had to supply them with energy. We had to increase their energy from this level to this level until we reach a level where their energy is capable of making them collide together and form bonds to produce the products. This is the blue curve, as you can see here, represents the uncatalyzed reaction. I mean, it represents the reaction without enzymes. So it has a relatively high level of activation energy because the level of the reactants was this, and we had to increase it until we reach that peak, which is the energy that we need to start with in order to start the chemical reaction and in order for the reactants to produce the products. The first remark here is that we have two curves. The first curve is blue in color, and it is the uncatalyzed reaction. And there is another curve, the green curve, that represents the uncatalyzed reaction, or that represents the reaction, the activation energy without enzymes. So of course, if this dotted line, if this, I will be changing the color. Now, this is the original level. This is the original level of energy for the reactants. And you need to give them energy to start colliding with each other and giving products. In case of the catalyzed reaction with enzymes, you need only this amount in order for the reactant to give the products. But in case of the uncatalyzed reaction, you need a surplus of energy. You need to raise the energy more for the reactant to start. So we can say that the activation energy in the uncatalyzed reaction is much more than the activation energy in the catalyzed reaction or when there are enzymes. So this is the first important conclusion we can get from the graph. The second conclusion here is that there is a difference in Gibbs free energy, delta G. Delta means difference, and G is Gibbs free energy. So delta G is the difference in energy or difference in free energy. You can see that this red arrow represents the difference in energy between the reactants and the products. Here, it's clear that the energy of the reactants is much higher than the energy of the products. The energy of the reactants stands for reactants is greater than the energy of the products. This means what? This means that there is a difference in energy that will be dissipated, that will leak to the environment. And if you're holding the test tube, you will find that the test tube is getting warmer or the test tube is getting hotter. Why? Because when the reactants produce the products, they release heat. So the reaction releases heat, and therefore this is an exothermic reaction. We call this exothermic reaction. So what is a exothermic reaction? An exothermic reaction is a reaction that is associated with release of energy. And therefore, an exothermic reaction is a reaction where the energy of the reactants is greater than that of the product. So again, an exothermic reaction is a reaction where there is release of heat to the environment. Why is there release of heat to the environment? Because the energy of the reactants is greater than the energy of the products. Of course, not all chemical reactions are exothermic reactions. There are some reactions that are endothermic reactions, meaning that if I have two substances, A and B, in a test tube that are reacting with each other, instead of releasing heat, they absorb heat in order to give a product C. So the reaction is no more called exothermic reaction, but it would be called an endothermic reaction, meaning a reaction that absorbs heat from the environment. Where does this heat go when A and B react together? It is given to the bonds in C, and therefore the energy of the product C will be greater than the energy of A plus B, meaning that the energy of the products is greater than the energy of the reactant. Of course, in such a case, the graphical representation would be like this: time here, energy here, reactants, level of energy of the reactants here, again, activation energy, but the energy of the product this time will be higher than the energy of the reactants. And now we have bypassed or surpassed all the obstacles and the difficult theoretical explanations, and we move on to the most interesting part, which you all like, and which I also like to explain, which is the part on the anatomy and how it is related to the diseases and the disorders. We talked before about mechanical digestion, and we said that we break down large food particles into smaller ones using physical force, and that is important to obtain a greater surface area for enzyme penetration, meaning that mechanical digestion, in brief, we can say that mechanical digestion prepares for chemical digestion, And we said, you gave an example, the famous example of chewing by the teeth, because chewing using the teeth, whatever the type of tooth is, helps in particulation or conversion of large particles into smaller ones. But actually, the mouth, the oral cavity, is not the only place where mechanical digestion occurs, and the teeth are not the only organs or structures that help in mechanical digestion, but we can find it all along the digestive system, or actually the alimentary canal. And another last property of mechanical digestion that is very important is it does not involve chemical reactions. What other type of mechanical digestion are we going to talk about? It's mechanical digestion, that's a bit weird. It's mechanical digestion carried out by the muscular wall of the alimentary tract, and it is called peristalsis, and we will be explaining it. So we will find it in the stomach, we will find it in the small intestine, we will find it in the small intestine, in the large intestine, because all these organs or structures have muscular walls. And by the way, we say smooth muscles because smooth muscles look like this under the microscope, and we will be seeing them under the microscope. They are smooth sheets of very delicate muscular cells, and they are not striated under the microscope, because you know that the skeletal muscles are striated. They are not striated, and most importantly, they are involuntary muscles. You can give an order to your biceps muscle in the upper arm to contract and move your arm, your biceps muscle, or your triceps muscle behind. But you cannot give orders to the muscles in the esophagus or the muscles in the wall of the stomach to contract. That's why we call them involuntary. So you cannot have control on the muscular movement of your digestive system or the alimentary canal. And we will be coming very soon, or moving very soon, to peristalsis and how peristalsis works as a way of mechanical digestion. So up till now, we talked about chewing, that is only in the muscle, and we will be talking about peristalsis, which is all along the alimentary canal. So these are the two major mechanisms of mechanical digestion, One of them is carried out via the teeth, and the other one is carried out via the muscles. But before doing this, we will have a rapid review of chemical digestion, which comes after mechanical digestion. And we said before that the type, hydrolysis is the type of chemical reaction that is used in digestion. So if I ever tell you or ask you, what is the type of chemical reaction that helps chemical digestion? Is it neutralization? Is it precipitation? Is it substitution? Is it hydrolysis? It is hydrolysis. And as you know, it is the breakdown of molecules using water. So here we give you an example. You needn't, of course, memorize the structure or be able to write the structural formula or draw the structural formula of sucrose. Sucrose, as we said before, is the common table salt, and it consists of two sugars that are very close to each other, which are glucose and fructose. And as you know, they are bonded together to form this disaccharide. And when we say disaccharide, as I said before, saccharide means sugar and di means two. So it's a rather large molecule or big molecule of sugars, of carbohydrates, that consists of two smaller sugars. Each one of these is called a monosaccharide. So again, glucose and fructose are both monosaccharides, meaning that they are the simplest known sugars. But here, they are bonded together via a bond. You needn't know the name of the bond, but it's a glycosidic bond. glycosidic bond. It's an oxygen that is connected via two single bonds to two sugars. So you have a sugar here and a sugar there, and they form together that big molecule, which is called the disaccharide. Why di? Because it's made up, the molecule is made up of two sugars. When you add the water, water is H2O. If you split the water, so it is H and OH. So actually, when you add water, you're adding H and you're adding OH. So here we add the H, there we add the OH, and now each of the two molecules is stable and no more needs the reaction, so they split from each other. They no more need this bond. And this explains to you how a molecule of water can split a disaccharide molecule. And now we move on to the anatomy inside the mouth cavity and the teeth. And we have several types of teeth, and we can classify the teeth according to either their shape or according to their characteristics and the age at which they appear. Okay, so first of all, according to their shape, we divide them into the incisors, which are the four front teeth. The four front teeth are called the incisors. And then we have on each side of the incisors, we have canines, which are usually more, the incisors are sharp, flat teeth like this, and they are four in number, four in the upper jaw and four in the lower jaw, and they are used to cut food because they are sharp or to bite. But we have the canines on each side. The canines are rather pointed, and some people have rather longer canines than others, or sometimes shorter. They are pointed teeth, pointed. They are very well developed in carnivores. Of course, if you have a cat, for example, if the cat opens its mouth, you will find two prominent canines like this, or in lions and tigers, which all belong to carnivores. Carnivores eat meat, they eat flesh, and it's important for flesh tearing. And when we say tearing, it's cutting with pulling. Of course, you saw, you might have seen a cat or a lion or a tiger like detaching meat from a piece of bone, or detaching meat from a dead animal, a dead body, and how this occurs mainly via the canines, which are the most prominent teeth in the mouth of such carnivores. And then we have, after this, the premolars and the molars. The premolars and the molars are broader teeth. The premolars come first and then followed by the molars. Of course, their presence and their number differs according to age, as we are going to see later on. So if this is a set of adult teeth, how did I know? From the number, because we have 10 teeth per jaw, 10 teeth above and 10 teeth below. Then these are adult teeth, or an adult set of teeth, and therefore they are permanent teeth, meaning that they have to stay in your mouth forever unless they get diseased or broken down. So the first one after the canine is the premolar, and then the rest are the molars. And they are, if you look at them from the underneath surface, you will find that they are a bit split, but they are broad and heavy for grinding food, okay? Another way of course, here, this is the picture of a child who has his teeth being changed. Of course, here is the gap that is left in the gums after the tooth has fallen, and the tooth that fell down and was lost is called a milk tooth. This is in opposition to the teeth that we saw on the previous slide, and that are called the permanent teeth. So according to their durability, we divide them into deciduous teeth, like deciduous trees. You know, the deciduous trees are the trees that lose their leaves in autumn or in fall. So deciduous means lost or shed away. So these are teeth that are being shed away at a certain age, and they are replaced by permanent teeth. We sometimes call them milk teeth because they are very white in color and because they appear in young kids that maybe are still milk fed, but mainly because of their white color. So they're so called because they're lost and they are replaced by other teeth, and they are called white teeth because they are white in color. And they become replaced by the permanent teeth, as I said before, that are supposed to stay in place unless they get loose because of bad oral hygiene. And here we have a rapid overview of the teeth. Of course, these are the milk teeth, and they are only here, let's count the number of teeth. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So 10 is the number of milk teeth. So actually, it's not here on that slide. I think this is a permanent set of teeth. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 16 per jaw. This is, sorry, the number of permanent teeth. I apologize again. So 16 per jaw. This is the number of adult teeth or permanent teeth, whereas 10 only per jaw is the number of milk teeth or deciduous teeth. So here we have 10 per jaw. We have the 4 central incisors, and then we have canines, and then we have 2 molars. So the molars are only 2, and there are no premolars, and these are the major differences. So we have 3 differences. The first difference is that we have only 10 teeth per jaw, and this is because there are no premolars, there are no premolars, and there are 2 molars on each side per jaw, only 2 molars. But let's look at the permanent teeth. In the permanent teeth, we have 16 teeth per jaw. And these 16 teeth per jaw are what? Consist of what? They consist of the 4 incisors. We have central incisors and lateral incisors, and have canines on each side. And then we have the premolars and the molars. The premolars that weren't at all figuring in the primary set or the milk set of teeth are present here. We have 2 premolars on each side, and then we have 3 molars. The first molar, the second molar, the third molar, and of course you can go over the age at which each of the permanent teeth is erupted, appears, or the age at which the milk teeth are erupted. And as you can see, all the milk teeth erupt. The age is in months because the maximum age is 33 months. 33 months, this makes about 3 years old. So by the age of 3, the child should have finished the set. All the milk teeth should have appeared. But on the other hand, the permanent teeth, if we look at the permanent teeth, the maximum age is around 13. The second molar appears at 13, but the third molar, which we call sometimes ders al-aql in Arabic, is or appears at the age of 21, and sometimes does not appear at all. And now we come to the tongue, and the tongue is, of course, a muscular structure. It belongs to the muscular system, but it's also common with the digestive system. But essentially, it is a muscular organ that contracts and relaxes. And when it contracts and relaxes, it moves. So it is a movable organ. So the nature of the tongue is muscular, but the tongue is used in speaking, of course, speech, speaking. It is also used during chewing and swallowing. It is also used during chewing and swallowing because it moves the food inside the oral cavity to allow the particles of food to aggregate together for easier chewing and for easier swallowing, but it is also important for taste. We said that it is movable and it helps in speaking and chewing and swallowing because it is a muscular organ, but it also contains nerve endings, sensory nerve endings that help the tongue to taste. So there are sensory nerve endings in the tongue. Of course, there are sensory nerve endings for heat and cold, so you can feel if the food is very hot or if the food is cold, but there are also sensory nerve endings for taste. There are sensory nerve endings for pain if the tongue is injured. These sensory nerve endings are responsible for the main tastes that we can feel by the tongue. The bitter taste is at the posterior most part of the tongue. So if you swallow a bitter medication or a pill that has a bitter taste, then you will mostly feel the bitter taste at the back of the tongue. And this is very important. When I say that this area is for bitter, that those two areas are for sour taste, for detecting sour taste, those two areas at the edges of the lung for detecting salty taste, and the tip of the tongue is for detecting sweet things or sugary things. I mean, where they are best detected. So the bitter taste is best detected at the posterior most part of the tongue. The sweet taste is better detected at the anterior, or mostly detected at the anterior most part of the tongue, or what we call the tip of the tongue. And then we have the sour taste and the salt taste, which are detected laterally at the edges. So the salt is lateral and anterior, and the sour is lateral and posterior. In addition to this, there is a taste that, of course, when I say taste buds, I mean the sensory nerve endings. Any nerve, if this is a nerve cell, for example, a sensory nerve cell with its dendrites, the neuron, as you know, and it has a long axon, okay? But at the end, you can see a modification, a change in shape to allow it to carry out the function of taste. So the taste buds are nothing but modified in shape and in function, of course, modified sensory nerve endings. In addition to the taste you already see on the diagram, there is a taste that's widespread on the central part of the tongue, and that has rather recently been described, which is called the umami taste. Umami taste. The umami taste is actually the taste of the proteins that are present in soup and those that are present in cheese. And so the umami taste is actually the taste of anything that greatly stimulates salivary secretion. If you review the types of food you eat, you will find that the greatest stimuli for your salivary secretion is through these umami substances that contain proteins and certain other substances that greatly stimulate saliva or salivary secretion. And that's why people like proteins or meat and cheese in general, because when something stimulates your salivary secretion, it makes you feel the taste more. and that's why we like eating soup or eating cheese, most of us, of course, and proteins like meat and chicken. In the mouth, mechanical digestion and chemical digestion occur together, but of course, mechanical digestion occurs first, followed very soon by chemical digestion. And actually, the main helper of chemical digestion in the food is the salivary glands, because they produce saliva, and saliva contains a very important enzyme called salivary amylase. And saliva is secreted not only the trigger, what is the trigger or the stimulus for secreting saliva? The trigger or stimulus for secreting saliva is not only putting the food in your mouth and tasting it, but even smelling food, or even seeing food, or even remembering a type of food that you like, will stimulate or reflexly stimulate salivary secretion. So the salivary secretion is a reflex. It's an involuntary response. Reflex salivation. Reflex salivation. Okay? And saliva is a mixture of things. It's a mixture of water. And by the way, most digestive juices, like saliva, it's a digestive juice, by the way, digestive juice. Most digestive juices contain water. They contain 90% water, and this is helpful for hydrolysis. But it also contains a very important enzyme whose function will be discussed very soon, which is amylase, and we call it salivary amylase because there is another amylase enzyme secreted by the pancreas. And there is also mucus, and mucus is a protein that is present in the nasal cavity and also present in saliva. But in saliva, it's dilute mucus. It's more dilute than in the nasal cavity, of course, and it is important for many things. First of all, it helps to stick the food particles together in order to allow for swallowing. And it also lubricates because it's slippery. It lubricates also to facilitate swallowing. But moreover, mucus is slightly alkaline, and therefore it is protective to the teeth, because the teeth are injured by acid, and all the holes and all dental caries, which are the cavities inside the teeth that are formed due to improper tooth hygiene. Dental caries are cavities, are due to acid produced by the bacteria in your mouth. So mucus, by being slightly alkaline, protects the teeth against tooth caries or cavities. What about the salivary amylase? It acts mainly upon carbohydrates. It helps the digestion of starch. Of course, you know that bread is made of or out of flour, and flour is a polysaccharide because it contains starch. So the flour that's used in making bread is a polysaccharide, and it is an important polysaccharide taken from wheat that is called starch. So your bread is full of starch, and salivary amylase catalyzes this reaction. It catalyzes the breakdown of starch or the digestion of starch using water. So it catalyzes hydrolysis of water. So the chemical reaction is catalyzed by salivary amylase. This is the enzyme. We need water, of course. We need the substrate, which is starch. Starch is the substrate. And we have a product, which is the disaccharide maltose. Maltose is a second example of disaccharides. We saw sucrose in the previous slides, which is the common table sugar. and now we see maltose. Sucrose was made of two sugars, which were glucose and fructose. Maltose is made of glucose and glucose. So maltose is purely glucose. Every two glucose molecules that bind together will form the disaccharide maltose. And that's why there is a famous question, but easy question, that I always ask you, which is, why when we bite with the first bite of bread or a croissant or whatever thing that contains starch, bakery in general, you feel a sugary taste in your mouth? You feel a sugary taste in your mouth because the bread contains starch, and starch is hydrolyzed using water and in the presence of salivary amylase to give the sugar maltose. Maltose is a disaccharide. It's not the common table sugar, but it's sugary. Here we can see several examples of polysaccharides actually. The one that we are interested in is the starch, because starch is digested into the disaccharide maltose, and the disaccharide maltose contains two molecules of glucose that are bonded together. Starch is the storage form of carbohydrates in plants. So inside a potato or inside a sweet potato, the extra carbohydrates are stored as starch. Cellulose is another carbohydrate, another polysaccharide, but it is present only in the cell walls of plant cells, in the cell wall of a plant cell. Glycogen is the most complex, as you see, branched, highly branched polysaccharide, and glycogen is the storage form of carbohydrates in animals, like human beings, for example, animals or human beings. So give me an example of an organ in the human body that stores glycogen: the liver. Whatever surplus of carbohydrate that you eat will be stored as glycogen in your liver, and it will also be stored in your muscles. Your muscles will use the stored glycogen to produce energy during muscular exercise, and the liver will use it whenever you are fasting, because glycogen can be broken down to glucose, or to maltose first and then to glucose, that might reach the blood during fasting or during starvation. Of course, we talked before about the pharynx, and we said that the pharynx is a cavity or an open area at the back of the mouth, and it serves as a passageway for both the digestive system and the respiratory system, because food air first can pass from the nasal cavity into the larynx, but through what? Through the pharynx. Similarly, food can crisscross and pass to the esophagus, but again through the pharynx. So the pharynx is a common cavity for both air and food. When we chew food, food is mixed with saliva and it becomes converted into a soft, rounded thing like this, which we called the bolus of food. So the food is first chewed and then it's moistened, of course, by saliva that contains mucus, and then it's rolled into a bolus, and finally there will be swallowing. And as you know, swallowing is a reflex action. We talked about the epiglottis, and we said that the epiglottis is protective against choking, because during swallowing, which is a reflex, there is another reflex, which is the downward movement of the epiglottis. As you see here, the downward movement of the epiglottis, it will move like this in order to seal the glottis, or the opening of the larynx, and protect against choking. And by doing this, it does not only prevent food from entering the trachea, but it also helps the food to pass into the esophagus. And now we come to the esophagus, and the esophagus, as you know, is this muscular tube that connects the pharynx to the stomach. The esophagus starts in the pharynx, at the bottom of the pharynx, the lowermost part of the pharynx, which is the laryngopharynx, and then descends through the neck and then through the chest cavity, and then passes across the diaphragm to reach the stomach. So it actually connects two structures. It connects the laryngopharynx to the stomach. And as we said during our explanation of mechanical digestion, all the organs, starting from the esophagus to the anus, have muscle layers. And these muscles are smooth muscles, meaning that these muscles are involuntary muscles. We do not have control on them. And the muscles in the esophagus are formed of two layers. So if this is the esophagus, the wall, then we have longitudinal muscles like this, where the muscle cells are elongated, and this is outer. So we have an outer longitudinal muscle layer, of course, smooth muscle layer. And then if we cut a longitudinal section of the esophagus to see what's inside, we will find an inner layer where the muscles are circular. So we have an inner circular layer. So circular, if you see the whole thing, circular will be like this, like rings, one above the other. But here, because we had a cut in the wall of the esophagus, so the outer layer, as you can see here, these are the muscle layers. See how the fibers run parallel to each other and longitudinal. This is the outer longitudinal layer of muscles. And on the other hand, we have another inner layer, as you can see, where the muscles are circular. And here we have made a cut on the inside in order to see the circular muscle layers. But again, you can see that there are nerves and blood vessels in the wall of the esophagus, or in the connective tissue around the esophagus. So this is the muscular wall of the esophagus, and it is this muscular wall that allows the esophagus to carry out peristalsis. And peristalsis, as we said, is the second example of mechanical digestion, which is broader than, or more extended in the digestive system, because it is all down the alimentary canal. And peristalsis is nothing but regular, rhythmic means regular, at regular intervals, contractions and relaxations. The aim of peristalsis in general, which consists of contractions and relaxations that alternate with each other, the main purpose is mechanical digestion. But mechanical digestion through what? First of all, by compressing the food. When the muscles contract, they compress the food and they make the food be converted into smaller particles, and then mixing the food with the digestive juices, but more importantly, sweeping down food or pushing down food. So one of the major, of course, pushing down is not related to mechanical digestion, but compressing and mixing food, it's like a mixer effect. Of course, with different degrees. The strongest peristalsis is done in the stomach. The strongest peristalsis is obtained where? Is obtained in the stomach. And needless to say that it is obtained in the stomach because the stomach has the strongest wall. The strongest muscular wall is present in the stomach. To tell you how strong it is, I will just say that it consists of 3 layers of smooth muscles, not only of 2 layers. So peristalsis is very important to push down food. And we see here, how we have a bolus of food. This is the bolus of food. And then the arrows that are directed inwards mean that we are compressing the esophagus above, while the muscles underneath are relaxed, and therefore the lumen is wider. So you have something that's narrowing here and something that's dilating here, and this helps pushing the bolus of food downward. And when the bolus of food is pushed downward, that part that was relaxed will now constrict or contract. Constrict is not contract. Constrict is the result of contract, because constrict means getting narrower. Okay, so when the muscle contracts, the esophagus constricts. And when they relax, they dilate, they get wider. So here you have constricted esophagus because of contraction, and here you have dilated esophagus because of relaxation of the muscles. The stomach, as we said before, we all know, of course, that if this is the abdominal cavity, this is the diaphragm, this is the umbilicus, and this is the groin. If we divide the abdominal cavity into 4 quadrants, then this would be the left side of the person in front of us, or the patient. So this would be the left upper quadrant, as we saw on the 3D model, and the left upper quadrant, the left upper quadrant is here, the left upper quadrant. So the left upper quadrant is the place where the stomach is present in the abdominal cavity. And as you can see, the stomach consists of, or the wall of the stomach consists of 3 layers: an outer longitudinal muscle, and the circular muscle, which was inner, will now be considered the middle layer, will now be considered the middle layer, whereas the innermost layer is an oblique layer of muscle. Oblique means that the muscle cells are directed like this. Again, we can see here the middle circular layer, the inner the inner oblique layer, and the outer longitudinal layer. It starts oblique and then it descends longitudinal like this. But through this diagram, we can also see the parts of the stomach. The first part of the stomach, which is just under the esophagus, is called the cardia. The second convex top of the stomach is called the fundus. And then the greatest majority of the stomach is called the body. And then we have the last part, which is called the pylorus. So we have the cardia just under the esophagus, the fundus, which is the convex top of the stomach, the majority, which is the body of the stomach, and then the last part of the stomach, which is called the pylorus. But we can also see that the stomach has 2 curvatures: an outer curvature that is called the greater curvature, and an inner curvature towards the middle line of the body, which is called the lesser curvature. What you see inside is the endothelium of the stomach. Here we have cut a part of the stomach in order to see the inside, and this is the endothelium of the stomach. This is the endothelium of the stomach, and it has folds like this, which we call the rugae, because ruga is 1 fold, rugae, and they increase the surface area. We will stop here and we'll be continuing in the classroom next week, inshallah. Thank you, guys.